CHEM 313, Lecture 14. So to review from Lecture 13, we introduced our unit on metabolism, and specifically we're emphasizing aerobic respiration. So you'll recall that aerobic respiration is the process by which carbon-rich fuels are oxidized to give us carbon dioxide. The electrons from those fuels are going to reduce electron carriers. The two most important are NADH and FADH2. When they don't have the extra electrons, they're called NAD plus or FAD. And those electrons are then going to be used to reduce oxygen. And that releases energy, and the energy can be used to power ATP synthesis. So what we talked about last time was we introduced the topic by introducing glycolysis, which is the first steps in using glucose as a fuel source. So that means that today we're going to take a look at the next steps in glucose as a fuel source. And also we're going to be looking at fatty acids as a fuel source. All right, we showed this slide last time. This slide shows the process of going from fuels to building blocks down to this key critical metabolite called acetyl coenzyme A, which is an easily oxidized molecule, and we oxidize it to CO2, and that leaves us with electron carriers, and the electron carriers can then be used to reduce oxygen. So up here we have oxidation of carbon, and down at the bottom we have reduction of oxygen and release of energy to give us ATP. So last time we covered glycolysis. Okay, we didn't completely close in on acetyl coenzyme A last time, but we did talk about glycolysis, which is the process of going from glucose to 2 molecules of pyruvate. And the pyruvate gets made into acetyl-CoA. So how that works we're going to talk about today. Once the acetyl-CoA is formed, we can oxidize it in the citric acid cycle and then feed electrons into the electron transport chain, which is how we power ATP synthesis. We're not going to get there today, but what we are going to get to today is the use of fatty acids as a fuel source through this process called beta oxidation. And beta oxidation converts fatty acids directly into acetyl-CoA without the need for going through glycolysis, right? But before we get to that point, I'm going to recap glycolysis a little bit just so that everybody is on the same page for what's really important to know about glycolysis. Okay, so let's review glycolysis. All right, so there are some things that you need to know about these biochemical pathways, where and when they happen. So glycolysis happens in the cytoplasm of all cells whenever ATP is needed. So in other words, you're working out or something and your ATP levels are low, or glucose is abundant. The purpose or the overall goal, or the why of this pathway. We said there are two of them. The first is to generate a small amount, some ATP, without any oxygen being involved. The second is to generate pyruvate for further oxidation. Okay, so we have to talk about what are our reactants and products, what are our inputs and outputs. We said that we have glucose plus 2 ADPs plus 2 phosphates plus 2 NAD plus reacting to give us 2 pyruvate, 2 ATP, and 2 NADH. And then we talked about regulation. So the main means by which glycolysis is regulated is by allosteric regulation of 3 steps. The first one is step 1, which is glucose reacting to give us glucose 6-phosphate. And that requires ATP, so ATP gets converted into ADP. The enzyme is called hexokinase. And we said that glucose 6-phosphate, the product of this step, inhibits this step. So as soon as the product builds up, the reaction goes slower. The second reaction that's regulated is reaction 3. Fructose 6-phosphate converted into fructose 1,6-bisphosphate. ATP is used up in this reaction. The enzyme is called phosphofructokinase. And we said that phosphofructokinase is inhibited by ATP and activated by a molecule called fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is not the same thing as fructose 1,6-bisphosphate. It's made separately from glucose, and its entire function is just to allosterically activate phosphofructokinase. The third regulated step I'm going to put up above because I'm running out of space. That's reaction 10 of glycolysis. That's when we take this intermediate called PEP, phosphoenol pyruvate, convert that into pyruvate, and in the process ATP is synthesized. The enzyme is called pyruvate kinase. Pyruvate kinase is inhibited by acetyl coenzyme A, and it's activated by fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is the product of reaction 3, so it activates reaction 10. Okay, so knowing these 3 reactions are the regulated steps, that's important, but the key question now becomes why? Why these 3 steps? Well, we said last time that reaction 3 is the rate-limiting step of glycolysis, and it's also the committed step. So what do those mean? Rate-limiting step means that it's the slowest step of glycolysis. So this reaction sets the rate for all of glycolysis. So if you speed this reaction up, you speed up glycolysis. If you slow this reaction down, you slow down glycolysis. So it's a key control point. It's also a committed step, which is something different. So a committed step is a step whose product is not useful for anything except making pyruvate. So the earlier intermediates in this pathway could be used for other things, but fructose 1,6-bisphosphate and everything after that is not useful for other things, for the most part. So for that reason, if you carry out this reaction, you are basically committed, right? You are stuck with this fructose 1,6-bisphosphate, which is not useful for anything else except for allosterically activating the last step of the pathway. There's another reason that these three reactions are regulated, and that's something I did not mention in the last lecture, and that is that these three reactions are all irreversible. And remember, when we say irreversible in this class, what we mean is the delta G is very negative, so these reactions tend to be super favorable. All right, why does that mean we want to regulate them? Well, because they're so favorable, if we don't regulate them, they're going to happen whether we need them to happen or not. There's no way to make these reactions run in reverse or stop. So it's not always the case that irreversible reactions are regulated reactions, that there are plenty of regulated reactions that are not irreversible. There are plenty of irreversible reactions that are not regulated. But irreversible reactions are more likely to be regulated than reversible reactions because you can't undo them once they've happened. Now, why these molecules? Well, we said before, ATP is a product of glycolysis. And so if we have plenty of this product, why do we need to carry out glycolysis some more to make more of a product that we've already got plenty of, right? Likewise, acetyl coenzyme A. So acetyl coenzyme A is not a product of glycolysis, but pyruvate is eventually made into acetyl coenzyme A. So it's the same sort of deal, right? The product of a pathway tends to inhibit that pathway to prevent waste. And as for activating the pathway, look at this. Fructose 1,6-bisphosphate activates reaction 10. Well, that's because we said fructose 1,6-bisphosphate is a useless otherwise product, and it's an intermediate that comes before reaction 10. So if fructose 1,6-bisphosphate levels build up, we can activate the last step of glycolysis and make sure that our fructose 1,6-bisphosphate gets used up. So there's a general principle that's true of metabolic regulation. It's not a hard and fast rule. There are exceptions, but it's a good rule of thumb. And that is products of pathways tend to inhibit those pathways whereas reactants of those pathways or intermediates tend to activate those pathways. There are exceptions, but that's a pretty good rule of thumb. So the whole point of glycolysis is eventually generate ATP. Therefore, ATP inhibits glycolysis to avoid waste. And that's generally true of many, not all, catabolic pathways. All right, so after we complete glycolysis, we have made our 2 molecules of pyruvate. And in the process, we've generated 2 ATP. Now, we said one of the nice things about glycolysis is that glycolysis is essentially the only way to get ATP without any oxygen. And that's important, for example, if you're in muscle cells that are doing heavy work without adequate breathing, or if you're just at the beginning of your exercise, your muscle cells are going to have to get a lot of ATP to contract, and glycolysis is the only way that they can get ATP if there is no oxygen present in the cells, right? Because it takes a while for your circulation to get enough oxygen to muscles that are actively respiring. So glycolysis is a way that we can get ATP without oxygen, but this comes at a cost. So it turns out there's a big problem with using glycolysis by itself as a source of ATP. So one of the problems is you just don't get that much ATP out of it. You get 2 ATP for every glucose, which is hardly anything. But there's a bigger problem. The bigger problem is this. In most cells, NAD plus and NADH levels are very low. So the concentration of NAD plus and NADH together in total in most cells is on the order of 100 micromolar max. In some cases it's less than that. So other metabolites are at least an order of magnitude higher. So we're talking 1 millimolar or so minimum. So because NAD and NAD plus levels are low in cells, NAD plus is the limiting reactant, if you will, for glycolysis. And it gets depleted very easily. So you'll recall that NAD plus is one of the reactants for glycolysis. 2-NADH is our product. If our NAD plus supplies get depleted in cells, we can't keep glycolysis going because we're going to run out of reactants. Now, normally, this is not a problem if oxygen is present. And the reason for that is because NADH can be oxidized back to NAD plus with oxygen being reduced to water. That's the function of the electron transport chain, which we're going to talk about in a later lecture. But if oxygen is not present, this doesn't happen. So that means that if oxygen is scarce, we must regenerate NAD plus somehow. And the way that we regenerate NAD plus is by reducing pyruvate. And there's two separate reactions that are responsible for reducing pyruvate that we're going to talk about. One is called lactate or lactic acid fermentation. That's what humans do. And the other is ethanol fermentation, which is what yeast do. Okay, so our problem here is that if we are in low oxygen situation, our mission is we have to regenerate NAD plus, so glycolysis can keep going and giving us ATP. So what are we going to do? What we're going to do is we are going to reduce our pyruvate. And there's two possible reactions for doing this. The first is called lactic acid fermentation. Pyruvate reacts with NADH. NADH reduces pyruvate to form lactate, which is this molecule right there. So in other words, we convert the ketone to an alcohol. And there's an enzyme that does this called lactate dehydrogenase. And the point of this reaction, again, a lot of misconceptions around this reaction. The lactate is not the point. The lactate is basically a waste product. The point of this reaction is to give us more NAD+, which can then be fed back into glycolysis so that we can get more ATP in the absence of oxygen. The nice thing about this reaction is it's reversible, so that if oxygen becomes available again, you can just convert your leftover lactate back into pyruvate and then break down the pyruvate. So this is what happens in animal cells, and it's especially important in muscle. If you exercise without breathing properly, you're going to build up huge amounts of lactate in your muscle cells, which leads to soreness. And excessive lactate production is actually bad for your health because it's an acid. It's going to lower the pH in your cells. Cells tend to export it out into the bloodstream to get rid of it, but lowering the pH of your blood inhibits your blood's ability to carry oxygen, which just makes this whole lack of oxygen problem all that much worse. So yeast and certain bacteria have a different, somewhat more complicated way of dealing with this problem, and that's ethanol fermentation. So in ethanol fermentation, we have a two-step reaction. First of all, pyruvate loses its COO group in the form of CO2, and that gives us acetaldehyde. And then acetaldehyde can be reduced to ethanol by an enzyme called alcohol dehydrogenase. And that, in turn, regenerates an NAD+ that can go back to glycolysis and generate more ATP in the absence of oxygen. So this can be harnessed industrially, and this is why if you want good conversion of your yeast and your sugar to ethanol whenever you're making beer or wine, you have to keep oxygen out of the reaction mixture as much as possible. And that's because you want the cells to undergo ethanol fermentation instead of just breaking down the pyruvate to acetyl-CoA, which is what they would normally do. Okay, so this is done mostly in yeast and also in some bacteria. Okay, so sometimes you'll hear these forms of fermentation called anaerobic respiration. What we mean by that is just glycolysis plus some sort of fermentation, one of these two reactions, to regenerate your NAD+ so that you can use glycolysis for ATP. If oxygen is present, cells tend not to do these reactions. Instead, what cells do is they convert pyruvate into acetyl-CoA, which is then further oxidized in the tricarboxylic acid cycle, or the Krebs cycle. And this is a preferred mode of catabolism because it generates much more ATP. But it requires oxygen. So this whole process of going from pyruvate to acetyl coenzyme A is a little mini pathway known as pyruvate dehydrogenase. Pyruvate dehydrogenase is a complex of reactions that oxidizes pyruvate to give us acetyl coenzyme A and some other byproducts as well. So let's take a closer look at pyruvate dehydrogenase. All right, pyruvate dehydrogenase. Whenever you see this word dehydrogenase, you should be thinking oxidation. Okay, a dehydrogenase is any enzyme that catalyzes oxidation of a substance and reduction of an electron carrier. So pyruvate dehydrogenase does a redox reaction in essence. All right, so it's a gigantic multi-enzyme complex. This is a picture of what it looks like. And it's located in the mitochondrial matrix. So if we wanted to apply my criteria for biochemical pathways, where and when, this happens in the matrix of the mitochondrion. In all cells, when pyruvate is abundant, such as when glycolysis is running. And also oxygen, although oxygen isn't directly involved in pyruvate dehydrogenase. Okay, its purpose, or the goal, or the point, is generate acetyl coenzyme A, which is then going to be further oxidized for ATP. So our major reactants and products right there, pyruvate plus NAD plus coenzyme A react to give us acetyl-CoA and NADH and CO2. Pyruvate plus NAD plus coenzyme A, which I abbreviate as CoA-SH, because it has an SH group on the end of it, which is what holds your 2-carbon group. Those react to give us acetyl-CoA plus NADH plus CO2. Its purpose is to make acetyl coenzyme A for the citric acid cycle. Regulation, how is this regulated? We're not going to talk much about regulation of pyruvate dehydrogenase, but I will say that there is an enzyme called pyruvate dehydrogenase kinase, which phosphorylates pyruvate dehydrogenase. So you guys remember what protein kinases do, right? Such as pKa, cyclic AMP-dependent protein kinase. They change protein function by attaching a phosphate group to them. So pyruvate dehydrogenase kinase phosphorylates pyruvate dehydrogenase, and that inhibits this enzyme. And pyruvate dehydrogenase kinase is itself activated by cell signaling pathways, which we're not going to get into. Okay, so let's take a look very briefly at pyruvate dehydrogenase and what actual chemical reactions take place in this enzyme. All right, so pyruvate dehydrogenase involves 3 steps. You are not responsible for knowing each of the 3 steps, just knowing that it has 3 steps is enough. So the first step is that pyruvate reacts with this molecule called TPP, thiamine pyrophosphate, to give us CO2 and hydroxyethyl TPP. Pyruvate has 3 carbons, CO2 has 1 carbon. The hydroxyethyl group on HETPP is our other remaining 2 carbons. So what we do in the very first step is we just chop CO2 off of pyruvate. Step 2, HETPP reacts with a molecule known as lipoic acid, which is redox sensitive, and it's the oxidized form that reacts with HETPP. We end up with hydroxyethyl lipoic acid and get our TPP back again. All right, so we've recycled our TPP. The third reaction of pyruvate dehydrogenase is a 2-parter. First is the hydroxyethyl lipoic acid reacts with coenzyme A to give us our acetyl coenzyme A, which is our main product that we wanted, and our lipoic acid, but now in a reduced form. So in step 3b, our lipoic acid, which is reduced, is going to hand off its electrons somewhere. Where do you suppose the electrons are going to go? Well, this is catabolism, so I would say the electrons are going to go onto NAD+. We are left with our oxidized lipoic acid again, which can be reused, and NADH. So NADH, acetyl coenzyme A, and CO2 are our three major products of pyruvate dehydrogenase. The first reaction is the one that is blocked by pyruvate dehydrogenase kinase. So that is the regulated substep of the three reactions. Now, one thing about pyruvate dehydrogenase is that it involves a lot of essential cofactors, because this is a multi-enzyme complex and there's lots of unusual chemistry that takes place here. I'm not going to talk about the mechanisms of these reactions. I do that in the metabolism class, but not for this class. But I will say that there are some important cofactors involved in pyruvate dehydrogenase. So one important cofactor is thiamine, which is vitamin, I want to say B1. It's either B1 or B4. I can never remember off the top of my head. That is what TPP is made out of. So if you take a multivitamin supplement, that is the only thing that we need thiamine for, is to make TPP, which is used in pyruvate dehydrogenase and a couple of other random enzymes here and there. Second important cofactor is niacin. That, I believe, is vitamin B4. If not, it's B1. I may have the numbering on those two reversed. Niacin, I forgot to mention in the last lecture, is what NAD+ is made from. So you cannot synthesize NAD+ from scratch on your own. We need niacin to make it. A third cofactor that is needed for pyruvate dehydrogenase and also a few other enzymes is riboflavin, which is vitamin B2. So riboflavin is what the electron acceptor on FAD is made from. And it's also involved in the third reaction of pyruvate dehydrogenase. So that's what riboflavin is for. And a final cofactor that is necessary for the pyruvate dehydrogenase complex is pantothenic acid. So pantothenic acid is a B vitamin. I don't remember which B vitamin it is, and I'm not going to make myself look stupid by trying to guess it. But this is what coenzyme A is made out of, partially. So coenzyme A is essentially an adenine nucleotide with a pantothenic acid arm on it. It's a big complicated molecule, and it terminates with an SH group. And an SH group can react with a carboxylic acid to give you a thioester and water. So acetyl coenzyme A is actually a thioester. All right, so we've done glycolysis. Pyruvate dehydrogenase is an add-on to glycolysis. We've done that as well. And now we've made acetyl coenzyme A. So that summarizes how we get acetyl coenzyme A from glucose or from other carbohydrates. Now, glucose and other carbohydrates are not your only source of acetyl coenzyme A. You can also get them from fatty acids. So let's take a look at how we would get acetyl coenzyme A from lipids. And usually the way this is going to work is that triacylglycerols, remember that's the glycerol with 3 fatty acids on it, are going to be broken down into fatty acids, and then the fatty acids will be made into acetyl coenzyme A. And the process of going from fatty acids to acetyl coenzyme A is called beta oxidation. So let's take a look at that right now. But first, before we do that, let's take a look at how lipids are converted into fatty acids so that they can then be oxidized. All right, fatty acid catabolism. Fatty acid comes from two places. The first source is dietary lipids. So you eat something that has fats, it goes straight into your bloodstream, and you get triacylglycerols. Okay, great. The other main source of triacylglycerol is from internal stores in adipose tissue. So animals are great at storing triacylglycerol in adipose tissue. And so a majority of your fatty acids are going to come from adipose cells. And we already talked about what adipose cells are like. They're mostly this gigantic droplet of triacylglycerol with the cytoplasm of the cell just squeezed around the edges of it in the narrow space between the triacylglycerol and the plasma membrane of the cell. So we're going to talk for a moment about how triacylglycerols are broken down into fatty acids. So this process is under hormonal control. So that's a difference between fatty acid breakdown and glucose breakdown. Glucose can be broken down essentially any time that the cell needs it, as long as there's glucose present in the bloodstream. But it takes special hormonal regulation to break down adipose tissue. In other words, it takes cell signaling. So that's one thing to keep in mind. Another thing to keep in mind is that fatty acids, you recall, they're lipids, so they're not soluble in water. And that makes using fatty acids as a fuel source a lot more complicated than using something like glucose, which is water soluble. So here's how the whole process works. There's this enzyme called HSL, or hormone-sensitive lipase. So this is not the only enzyme that's involved in fat breakdown, but it's the most important one. So fatty acids are released from triacylglycerol by hormone-sensitive lipase. So hormone-sensitive lipase cleaves triacylglycerols and diacylglycerols into fatty acids. It just cuts them off of the glycerol. Now, hormone-sensitive lipase itself, the reason it's called that is because it's hormone-sensitive, surprise, surprise, and it has to be activated by pKa. That's the same pKa that we saw back in the talk about cell signaling. It's the one that is activated by cyclic AMP. So breakdown of fat, or triacylglycerol, from fat tissue requires cyclic AMP. All right, so we have our fatty acids. What ends up happening is the fatty acids pass through a channel protein, and then in the bloodstream they bind to serum albumin. So serum albumin is sort of an all-purpose protein. Its main function in this context is to carry nonpolar substances through blood. So fatty acid does not just float through blood because it's insoluble. It has to bind to the serum albumin. So what happens then is when the fat is taken to a cell that is using the fatty acid as energy, the serum albumin binds to a fatty acid transporter, which brings the fatty acid into the cell. And when the fatty acid is inside the cell, it immediately is attached to a coenzyme A. So what happens is a fatty acid plus coenzyme A react to give us what's called a fatty acyl-CoA. Fatty acyl-CoA is the same thing as acetyl-CoA, except in this case you've got a whole fatty acid attached to your coenzyme A instead of just a short little acetyl group. So that's what a fatty acyl-CoA is. The reason for that is because, for one thing, fatty acyl-CoA is more soluble than a plain fatty acid, so it can diffuse within the cell much more easily. A second, more important reason is that it is electrically neutral. If your cells started to take in a whole bunch of fatty acids are negatively charged, and that's going to screw up your membrane potential for taking things into the cells, and that's going to create problems. So fatty acyl-CoA is a way of hiding the electrical charge on a fatty acid. All right, so what we now have is fatty acyl-CoA inside our cells, and where those fatty acyl-CoAs are going to go is into the mitochondria, and specifically into the matrix of the mitochondrion. And so it does this through a complicated couple of steps of reactions. We're not going to talk about how it works, but fatty acyl-CoA goes into the matrix of the mitochondrion, and that's where it is actually broken down into acetyl-CoA. All right, so our next pathway, the beta oxidation cycle. Where and when? So this happens in the matrix of the mitochondrion. In any cells that can use fatty acids as an energy source. So the most important one of those would be muscle. Muscles are what use up most of our fatty acid. It does not occur in brain. Your brain cannot use fatty acids as an energy source. We're not going to talk about why, but that's an important point. When does this occur? This occurs when ATP levels are low, so you need more ATP. When glucose is not present, because glucose can be broken down by glycolysis much more easily. And most importantly, when oxygen is present. So beta oxidation absolutely requires oxygen. You cannot do this with there's no oxygen present. The purpose is to make NADH and FADH2, which can then be oxidized to give us ATP, and also to make acetyl coenzyme A, which is then fed into the citric acid cycle, and once again oxidized to give us ATP. So that's the purpose of the beta oxidation cycle. The regulation, so beta oxidation itself is not highly regulated. Okay, but release of fatty acids from adipose tissue requires hormone signaling. So that functions as a form of indirect regulation of when the cells break down fatty acids or don't. Okay, so for our major reactants and products, I'm going to save that for after I actually show you what the chemical reactions are that take place in beta oxidation. Beta oxidation is very, very simple. Unlike carbohydrate metabolism, it's just 4 repeating reactions. And those reactions are quite simple. Here's what happens. we start off with our fatty acyl-CoA, which looks something like this. And then what we're going to do is we're going to oxidize it and get FADH2. When you oxidize this, what we do is introduce a single double bond. So that's reaction 1. Form double bond and get FADH2. Next reaction, we add water so that we go from having a molecule with a double bond to adding water across that double bond, giving us a molecule with a hydroxyl on it. This is what's called a beta-hydroxyacyl-CoA. Third reaction. Third reaction is we oxidize the alcohol and give us NADH. So what happens is our beta-hydroxyacyl-CoA becomes oxidized to a ketone, and we get NADH as a byproduct. And then what happens in the fourth reaction is we simply cleave off the first 2 carbons to give us acetyl coenzyme A. So if you just cut this bond right there, what you're left with right here is an acetyl coenzyme A, which is released as a product, and then what's left over is the same fatty acid chain, except now it's 2 carbons shorter. And this whole process repeats over and over again until the entire fatty acid has been chopped up into these 2 carbon acetyl-CoA units. So that means that from our fatty acid of N carbons, we end up generating N over 2 minus 1 FADH2 and NADH, and we generate N over 2 acetyl coenzyme A. Or if you prefer the full notation, there it is down at the bottom left. Fatty acid plus N over 2 coenzyme A plus N over 2 minus 1 NAD plus FAD and H2O give you N over 2 acetyl-CoA and N over 2 minus 1 NADH and FADH2. This is for saturated fats. All right, for an unsaturated fatty acid, you subtract 1 FADH2 per double bond. And the reason for that is because of reaction 1. So remember, reaction 1 is the one where we form a double bond and get an FADH2 in the process. So if there is a double bond already present, there's no need for this step to take place, so we don't do it. It's just skipped over, and you lose out on an FADH2 in the process. So for that reason, unsaturated fatty acids give you slightly less energy than saturated fatty acids do. And once again, all of this requires oxygen. Without oxygen, you cannot break down fatty acids as an energy source. Okay, so what's going to happen next? Well, we have our N over 2 FADH2 and NADH. These go to the electron transport chain, which we'll talk about later. And we've got N over 2 acetyl-CoAs. These go to the citric acid cycle. And the citric acid cycle is where we get the bulk of our NADH and FADH2 that will then be used to reduce oxygen. So next time, we're going to talk about the citric acid cycle. So look forward to that lecture, and in the meantime, check out the homework.